The overall goal of this procedure is to present a complete and detailed process to apply RNA-seq, a powerful next-generation DNA sequencing technology, to profile transcriptomes. This is accomplished by first isolating total RNA from human pulmonary microvascular endothelial cells with or without thrombin treatment and checking the RNA quality. The second step is to construct DNA libraries from those RNA samples, streamline the cluster generation using the CBOT instrument, and carry out the DNA sequencing task on the HiSeq 1000. Next, data analysis is performed to ultimately identify and display differentially expressed gene transcripts. The final step is to validate RNA-seq results by RT-PCR.
The main advantage of RNA-seq over existing methods, like a DNA microarray for transcriptome analysis, is that RNA-seq can profile a complete transcriptome, provide digital type of data, and not rely on any known genomic sequence. Visualization of gene expression in cells while measurement of mRNA level is a useful tool in determining how the transcriptional machinery of the cell is affected by external signals, or how cells differ between a healthy state and a diseased state. In this protocol, we will demonstrate RNA-seq analysis of transcriptomes in Thrombin-treated and control human pulmonary microvascular endothelial cells.
This protocol is based on our recent published study in which we successfully performed the first complete transcriptome analysis of human pulmonary microvascular endothelial cells treated with thrombin using RNA-seq.